Living things have one of two different types of cells. They either have simple prokaryotic cells or more complex eukaryotic cells. Prokaryotic cells are usually smaller and simpler than eukaryotic cells. They have circular DNA, so their genetic material is arranged in a ring. They do not have a true nucleus or other membrane-bound organelles. Bacteria and archaea have prokaryotic cells, so we'll talk more about them when we talk about those groups of organisms. Here's another question for you from Participation Assignment 1. Eukaryotic cells are larger and more complex. All other life on Earth, including the protists, fungi, plants, and animals, have eukaryotic cells. In eukaryotes, the genetic material, or DNA, is linear and is contained in a membrane-bound organelle called the nucleus, and the cell also has other membrane-bound organelles. The membrane-bound organelles form compartments in the cell that are specialized for different functions. The nucleus is one of these membrane-bound organelles, which contains the genetic material in eukaryotes. Again, in eukarya, the DNA is in linear strands. So what is the significance of the evolution of the nucleus in eukaryotes? Because the nucleus is surrounded by a membrane, similar to the plasma membrane of the cell itself, the genetic material is more protected. The membrane allows the nucleus to control what molecules move into or out of it, and what comes in contact with the genetic material. Here's another participation question for you. Which of the following is true? Eukaryotes evolved from simple prokaryotes without a nucleus or other membrane-bound organelles. There are two theories for how early prokaryotes developed these membrane-bound organelles and became what we now call eukaryotes. Some of the organelles likely formed through the process described in invagination theory. Others we believe formed in a different way, which we describe using endosymbiotic theory. Invagination theory proposes that some membrane-bound organelles formed from infoldings of the plasma membrane. Many prokaryotes have these infoldings that increase the surface area of their plasma membrane. Invagination theory suggests that these infoldings surrounded some of the structures in the cell and pinched off from the plasma membrane, encasing those structures in a bubble of membrane. This formed these inner compartments that could become organelles specialized for particular functions. This is how we think the nucleus most likely formed in early eukaryotes. There are several other membrane-bound organelles in the eukaryotic cell that most likely were formed through invagination. The endoplasmic reticulum is an organelle extending off the nucleus that is important for the synthesis of new proteins. It contains the ribosomes, which are structures that build proteins using the DNA as a template. Remember that that is basically what DNA is, a recipe for making proteins. After new proteins are made, they go through some final processing steps in another organelle called the Golgi apparatus. Another membrane-bound organelle in the cell are the lysosomes. These are little sacs containing enzymes. These enzymes help break down molecules, either to digest food or to recycle damaged molecules in the cell. But some organelles in the eukaryotic cell, like the mitochondria and the chloroplasts in plants, are more complex than the other membrane-bound organelles. In fact, they seem to resemble small cells themselves. And in fact, the theory of how these organelles evolved suggests just that they were originally separate cells that merged with larger cells. This theory is supported by the fact that mitochondria and chloroplasts have many characteristics in common with prokaryotic cells. Firstly, these two organelles are similar in size to prokaryotic cells. Also, when a eukaryotic cell reproduces, the mitochondria and chloroplasts also divide in the same way that prokaryotic cells reproduce. This is a process called fission that we'll talk more about when we talk about bacteria and archaea. The mitochondria and chloroplasts also have their own ribosomes. Again, the ribosomes are structures in the cell that allow it to synthesize proteins. You've probably heard of mitochondrial DNA, so you know that the mitochondria of our cells have their own DNA separate from the cellular DNA. This is another reason we think the mitochondria and chloroplasts were originally prokaryotic cells. They have their own DNA, but it is circular DNA, like that found in prokaryotes. Remember that the cellular DNA in eukaryotes is linear. So how do we think this happened? Again, the endosymbiosis theory suggests that these organelles actually started out as separate cells that formed a symbiosis with larger cells. Endo means inside, so a larger prokaryote engulfed a smaller prokaryote, and the two merged together in a close symbiotic relationship. Over time, the smaller prokaryote lost some of its own functions and evolved into an organelle of the larger cell. The mitochondria are very important structures in the eukaryotic cell. You may have heard them referred to as the powerhouse of the cell. This is because the mitochondria are where most of the ATP is generated in cells. Again, we'll talk more about ATP when we talk about photosynthesis and cellular respiration. But this molecule stores the energy that cells use to do work, such as the active transport we talked about earlier. When we talk about cellular respiration, the process by which cells generate ATP, you'll see that it takes place mostly inside the mitochondria. The chloroplasts in plants, which we also think evolved through endosymbiotic theory, are where photosynthesis takes place. Now pause to answer this question from Participation Assignment 1. There are 2 more questions in Participation Assignment 1. Pause now to answer this question about chloroplasts. And finally, answer this question about the mitochondria.